Hello and welcome to the Geography Open Day Taster Lecture on geographical perspectives of malaria transmission. So my name's Dr. Mark Smith and I lecture on the physical geography side of the geography course. So that's my specialism is really looking at hydrology, how water moves through the landscape, but also geomorphology, and that's the shape of the landscape itself and the processes that form it. And for the last 10 years or so, I've been involved in projects that try and apply that knowledge, that classic physical geography knowledge that you might know from your A-levels or your GCSEs, to the problem of understanding malaria transmission in Africa. And I'd like to talk through some of that with you today. So I'll share my screen with you now and we'll go through the structure of the Open Day lecture. Okay, so what I want to do to begin with is just briefly give you an outline of what malaria is and what are the key questions we should be asking. So after that comes the main event where we look at ways that we can map and model malaria prevalence, initially at the continental scale, and that means Africa really, and then zooming in to see the real world complexity at the landscape scale. Now, I don't want to make too somber of a mood here, and I don't want to dwell on this, but we really have to acknowledge how terrible malaria is globally. This National Geographic statistic does need to be taken with caution, but the suggestion there is that perhaps 1 out of every 2 people who have ever lived on Earth has died of malaria. So that goes all the way back to cave people, etc. I think that statistic requires some caution, but you know, the fact that that's even being mentioned tells you how significant malaria has been to humans as a species. Perhaps more reliably and more recently, the World Health Organization's most recent report on malaria says that there are just over 405,000 malaria deaths in the world in 2018. And that's from 228 million recorded cases. Of those cases and those deaths, 93% are in Africa. So there's a reason for our focus here in Africa. It takes the majority of the world's malaria burden. Tragically, the demography of this disease is that it mostly attacks children. So of those recorded deaths, sadly, most of those will be children, simply because they haven't had chance to build up an immunity to malaria. And the people that live in a malaria endemic area are people that have survived their first bout of malaria. But there's better news that on the right-hand side, you can see the poster that tragically suggests that malaria kills 3,000 children every day. That was the case when that poster was made in 2010, but things have significantly improved since then, although the gains have actually slowed down in recent years. But how is malaria transmitted? How can we get malaria? And I think you probably know the essentials of this. The culprit is the Anopheles mosquitoes. So it's not all mosquitoes, it's just a certain type of mosquitoes, these Anopheles. And there's several hundred species of those. And so where are the secondary hosts? It's these mosquitoes that are the definitive or primary hosts. And malaria itself is a parasite, or 4 different Plasmodium parasites. And it's transmitted through the salivary glands of the mosquitoes. Prepare yourself for a terrifying diagram. Fortunately, this is not to scale, but it just is a really nice illustration of how the malaria cycle works. And when the mosquito bites you, it injects these sporozoites, these tiny little wiggly things, into your bloodstream. And eventually, they enter the liver and become a different form of the parasite called the merozoite. And when they get released from the liver, they attack the red blood cells, which you can see there, and they eventually differentiate into male and female forms that when the mosquito bites somebody a second time that has the malaria parasite, it's those forms that go back into the mosquito, eventually mutate in the mosquito's stomach, burst through the stomach wall, and make their way back to the salivary glands again. So the key thing is to note that while we often blame mosquitoes for giving us malaria, we actually gave it to them. It's a two-way relationship. Obviously, it's the mosquitoes that do the biting, but when it's born, a mosquito does not have malaria. It only contracts it from biting a malaria-infected human. So we're actually a pool of the parasite as much as the mosquito is. Understanding malaria is extremely complex. you can see on the left-hand side of the slide the number of things that we need to think about, moving from climate to the different parasites to the different vectors, that's the different species of mosquito, which all prefer different habitats. And then things like population, the economy, how much money does a society have to protect itself from malaria? And then things like culture, how much immunity is built up, etc. And there's this great quote from the 1930s saying that malaria is so molded and altered by local conditions that it sort of becomes 1,000 different problems and puzzles to deal with. So like chess, there's a few little pieces, but you can have an infinite variety of local situations that may require a locally specific problem. Now, I'm only going to talk really about the things in red on the left-hand side, but it's worth acknowledging these other layers of complexity. For example, I know a study in our field site talked to people about use of bed nets, and we discovered that if somebody had died and everyone went to the funeral, according to the local custom, would be at their house. And at that point, you would sleep outside of the house, and it would be viewed as rude to use a bed net before a funeral. Now, given that most people in that valley probably died of malaria, and so it suggests that malaria is bad at that location, if you don't include behaviors like that into your model or think about ways to try and educate, to inform those practices a little bit more, then you're going to miss a very important effect. So I've always thought of malaria as something that both human and physical geography can contribute to equally. But let's just focus on those environmental influences, those things in red at the top of the last slide. Two things really to bear in mind. Mosquitoes are dependent upon air temperature. The malaria parasite and the mosquitoes, the warmer it is, the faster these cycles go, where the little tadpole sporozoites develop in the mosquito, which they can then bite a human with. And actually, the warmer and more humid it is, the longer a mosquito can survive. In fact, because the mosquito needs to bite a human in order to get malaria, it's the older mosquitoes that you've got to watch out for. And it's only the females that do the biting. They bite to get a blood meal in order to lay eggs. And so really it's the granny mosquitoes that are the problem here. Not the young ones, not the male ones, it's the really old female mosquitoes, because they're more likely to have bit someone that has malaria already. So temperature is really important. And the other thing is water availability. You can see that actually that immature stage of the mosquito going from the eggs, which will be laid in a body of standing water, to the larval stages, to becoming a pupae before it disperses again, needs to take place in some standing water. So really it's temperature and water availability that are the two main environmental influences. So what we want to ask then is, where are these breeding sites going to form? What are the properties that makes a breeding site effective? Where will they form? And also, when are they going to be important? We need to do that in order to distribute often scarce resources and prepare as much as possible for malaria epidemics. And so effective targeting of our often limited resources is key here. So let's think about mapping where malaria is. The most obvious thing would be to take a map of places where people have malaria. But since the 1900s, we've been changing the environment to try and break the malaria cycle, to eliminate malaria from certain countries. So it may be that malaria isn't somewhere today in Italy, for example, not because it isn't climatically suitable for it, but simply it's been eradicated thanks to quite expensive interventions. So that limits statistical approaches, but we can still take what we might call a more biologically informed or a process-based approach, where we look at the fundamental controls on malaria transmission. For temperature, it's pretty well understood. You can see there's various curves on the bottom here. They've been informed from lab experiments, but the key point I want you to take away is the warmer it is, the more suitable it is for malaria, up to a point. So there's an optimum range, but to be honest, Africa's pretty warm already. So it's only the extreme north and south of the continent in winter and some high elevation areas that aren't warm enough. And you get an area in the desert that's too warm for a few months of the year. But actually, where that water is, we don't know that as well. And in fact, we use rainfall as a proxy, as a way of measuring water availability at the surface. So let's think about that. These same models that use these fancy temperature curves apply a monthly rainfall threshold, sometimes daily, but mostly a monthly rainfall threshold. What is that threshold? Well, let's take a range from 0 to 175 millimeters a month. The most commonly cited study, the Mara study, is around 80 millimeters, and that's quite a well-acknowledged threshold. Another threshold from Tanzas is 60 millimeters a month, maybe with a 1-month catalyst of 80 millimeters. And this is this threshold that we're going to focus on in future. But actually, you'll notice different studies over time have suggested different thresholds because they've been located in different places, and those places may be hydrologically very different. So unsurprisingly then, you get a range of thresholds depending on the environment in which they were produced, and funnily enough, depending on which threshold you choose, you get a wide variety in environmental suitability for malaria. So when you look at these maps, the rainfall threshold goes from left to right, 0 to 175 millimeters. And the more green a map is, the more months a year malaria is, the environment is suitable for malaria. So what can we do? Can we do this a bit more objectively? Well, over to you guys, really. You know, if this were a live lecture, I'd be asking you, what's wrong with just using rainfall instead of surface water availability? What hydrological processes have you studied during your GCSE or A-level years that you think probably should be involved in these continental scale maps of malaria? So I suggest pausing this lecture and just having a think about the things that you might want to add in to these models. So if you've unpaused now, what I'd say is that, of course, there are a lot of hydrological processes to include. We've got evaporation, where water goes back to the atmosphere. We've got infiltration, water going into the ground, uptake from plants, and of course, water runs off through rivers. Snowmelt, not so important in Africa, but in some places it is. And also irrigation practices might result in local water bodies. So what we did is we attempted to link this thermal approach, these well-known temperature curves, with a hydrological model that included all these processes. And that's the first time that's been done at a continental scale. Oh, lots on this slide, but let's focus on what is different. The top two panels on the left-hand side, A and B there, you can see on the left, we've got the standard classic rainfall threshold. And we can see the darker the green, the more malaria there is. And we can get an idea of the overall pattern. B is our new model with all the hydrological processes in place. And perhaps the first thing to see is that the overall pattern is quite similar. The extents are quite similar. But the more we zoom in into the detail, the more we see that it is actually quite different. First off, we can see the river corridors are highlighted very clearly. The Nile, the Niger, the Senegal rivers, some rivers in Somalia as well extend the geographical ranges predicted to be climatically suitable. Overall, we're seeing less overall area predicted to be suitable, estimated to be suitable for malaria transmission, but we're actually seeing more area being estimated to be suitable year round because we're routing water through rivers that are always wet enough. Adding irrigation, which is the bottom left panel, extends that further. So on the graphs on the far right-hand side, the green bars represent the area and population according to the rainfall threshold. The blue are those with our new hydrological model, and the red are when you add irrigation into that as well. And you can see with the irrigation, you get much more year-round suitability. There's not really a true data set that we can test this against because it's just climatic suitability for malaria transmission. It doesn't mean that it actually has to take place. But a good start is panel D and the bottom there, which is a map of malaria transmission in the pre-intervention era. So a sketch map really from the 1900s, very early 1900s. Generally, our model tends to overestimate malaria suitability in South Africa, but less so than the rainfall threshold approach. Conversely, we underestimate it in East Africa, and that's more pronounced than with the rainfall threshold approach. But why do we need to do this at all? Why would we care? Well, the problem is we need to understand what's causing the patterns we see today in order to predict them into the future and see what the impacts of climate change might be. So if we apply this model to climate change projections and compare it against the rainfall threshold approach, we can see quite a big variability. So that the blues here are where malaria is getting better, it's becoming less of a problem. The reds, it's becoming worse. And the more intense the color, the more our different climate models agree that it's getting better or worse. So this is up to 2100 under an extreme warming scenario. Both models suggest only a small increase in area, but we see that very different pattern. The estimates of increased malaria transmission suitability in South Africa follow a very different pattern. Rather than being focused in the east of the country, centered on Lesotho, the hydrological model actually predicts the affected area will stretch along the courses of the Orange River to the border with Namibia. And so this aridity-driven decrease in suitability across southern Africa that the rainfall threshold approach suggests, this green-blue area around here, isn't so pronounced in the hydrological model, whereas the projected decreases in West Africa are more pronounced in our hydrological model than with the rainfall threshold. But the largest difference by far is in South Sudan, where the hydrological model predicts a strong decrease in malaria suitability that just isn't observed in the rainfall threshold model. And then just briefly, I want to say that there's a lot missing in these continental scale models, and that when you zoom into the landscape scale, we see a lot more going on. This is based on some work that we did in Tanzania in the Kilimanjaro Valley. And we observed that essentially not all puddles are the same. You get some puddles that are formed in rivers, in riverbeds themselves. You get some puddles on the floodplains that form after floods. Some puddles are caused by groundwater springs, whereas others are based in sort of concavities in the landscape that form when the water table reaches the surface. So what we were interested in is seeing what makes a good larval habitat for mosquitoes. So we went out to this valley in the Kilimanjaro for a few years. We chose it because at the time it was one of the worst affected areas by malaria. They measure it in terms of the number of malaria-infected mosquito bites per person per year. And somebody observed, apparently, close to 3,000 bites. Now, I don't think that's too reliable, but it shows that it was a very severely affected area. And in the map in the bottom right, those white dots were areas that we wanted to sample. They covered a range of these water bodies. We thought, why has no one done this before? This is super important work. It's going to yield a lot of information on the processes that actually take place at the landscape scale. We soon realized why no one had really attempted it. It was very difficult to get to some of these places across a floodplain that's several tens of kilometers wide, and these were very long journeys to sample these water bodies. And then just briefly, what we found was that there were no significant relationships between larval counts and either rainfall in black at the bottom, or river level in grey, or in the water table depth in blue, the blue dashes. But when you break it down into those different water body types, you start to see some interesting relationships emerging. So actually, certain water body types turn on and off throughout the year, so the vector productivity shifts. Those areas of local saturation became productive when the river and water table was high, but those areas close to rivers had a bit of a lag of 1 month before that. But absolutely, our groundwater here was the star performer where most of the larvae were found. So just to summarize then, geography matters. Physical and human geographers can be useful in the study of malaria, even though the two things aren't often coupled together. And that continental scale models really need to improve and embed hydrology even more than this example has done, if we're going to really predict how climate change might affect malaria distribution. There's a lot more to be done on this. And even when you zoom into the landscape scale, you see that any attempt to do this at the continental scale makes big assumptions that actually may not be borne out at a local scale. We still need to have that local knowledge. Now, this is a taster lecture, so I mean, I don't want to go too far with this, but if you're really interested in this sort of stuff, there's a great book that's available, The Malaria Capers. It's an excellent readable introduction to this sort of thing, and it's better than more recent publications. And it's on Amazon, used from just £1. So it's within reach to maybe have a look at that if I've fed some interest in you there. So I hope that was useful for you as a bit of a taster lecture, and hopefully I will see you soon in a lecture theatre next year. Just to say, if you do have any questions about this presentation, I put my email address on that last slide, so do just skip back to there and feel free to email me any questions you've got. Thank you very much.